Hi, my name is Julie Snyder, and my partner Anna Roberts and I did our project on biofilms. So what are biofilms? Biofilms are protective shields made of carbohydrates, proteins, and DNA. They can prevent substances such as antibiotics from getting through to kill bacteria, which can be bad. But they can also do good things such as preventing harmful substances from getting through to kill good bacteria. Biofilms can also grow and mutate very quickly. So the Biofilms lab that we completed last year, we are going to complete again this year so we can become even more familiar with biofilms. In this lab, we use a diluted harmless bacterium called Pseudomonas fluorescens. This lab requires 4 steps that are completed over about a week's time. The first step of this lab is inoculation of the bacterium. The first day is taking a colony of bacteria and placing it into a culture tube with a bead and QB liquid in it. This is done twice for 2 separate experimental tubes. There's also a third tube that is a control tube that has bacteria but no bead in it. These tubes are labeled as day 1 tubes and then are incubated until day 2.
Step 2 is the bead transfer and plating, which starts on day 2 with the dilution of the bacteria. 3 new culture tubes are taken with a bead added to the 2 experimental tubes and QB liquid added to both the experimental and control tubes. A series of smaller tubes that can be mixed easily are set up for each experimental and control tubes. These will be used for the dilution. After adding the bead from each of the day 1 experimental tubes to the first microcentrifuge tube, a sample is taken for incubation and is labeled as day 2. The dilution series is then completed. After each dilution step, the tube must be vortexed. This happens 3 times with the last 2 dilutions being plated. For the control, the bead isn't transferred to the microcentrifuge tube. Instead, the liquid is. Here is a visual of step 2, where you can see the dilution process and you can see a sample being taken for incubation and the last 2 being plated. Step 3 is the bead transfer. 3 new day 3 culture tubes are taken with the bead added to the 2 experimental tubes and QB liquid added to both the experimental and control tubes. Day 2 culture tubes that were set aside the day before are taken out. The bead from the experimental tubes are transferred to their corresponding day 3 tubes, with just the liquid from the control tube being transferred. The same dilution series from day 2 is completed for days 3, 4, and 5. The last step of this lab is to examine your results. Compare your plates from days 2 and 5 and record the differences and similarities. Consider the size, shape, margin, elevation, and texture. Consider the differences between the experimental and control plates. Look at the percentage of day 5 colonies that look the same as day 2 and which look different. The ones that are the same can be considered the ancestral population, and the ones that look different are newer and have grown since then. Thank you for listening to our presentation. We hope that you enjoyed and that you learned something new.